Christophides is the other PI in this Transmission Zero project. And this is our generous funders. Thank you.
Thank you very much. And I think you stick to time. I forgot to start my timer, so thank you for that.
Okay, good.
Any questions? Yes.
Marty Rono here. Thanks for the nice talk. I've got a couple of questions. One, I've seen your phenotype looks like something I remember when you knock out a mosquito gene called lipophorin. The ovary reduction is hit, and also the parasite size become limited. And I think that was also done in Christophides' group. Is there anything that might be related to knocking out or introducing the antimicrobial peptides and affecting that pathway? And the other thing, do you modify the mosquito microbiome that might also affect other things?
So the microbiome we haven't looked at. I think this is something that George is looking now with a student. As to answer your question, it is actually the only extra slide I have. We did look at the CP locus that we're integrating. It is almost certainly not expressed to the same level as a wild type. So we did wonder whether this effect has something to do with the knockout of the CP. But we have used CP in another paper before with 3 different, no, actually, no, we've had different antimicrobial peptide, and we used it also for other things that are not published. And we haven't seen any effect from the CP knockout itself. And in fact, when we put a GFP in here to knock out the CP gene itself, surprisingly, it didn't actually have a big effect. We actually expected that a CP knockout would be pretty detrimental, but we did not. As you can see here, this is the oocysts barely affected. I think it's basically no difference. Also no different in the expert female. So we actually, in this experiment where we knocked out the CP, we didn't see any effect of the CP, any clear effect. Yeah, so I don't think it's the CP. I think it has to do with the antimicrobial peptides, yeah. Okay, yeah, I'm not familiar with those experiments, but yeah.
Thanks. While you're on that slide, so is that expected to make a null allele of CP, or is that in an intron, that GFP thing?
There's no intron. Okay, no, that's no intron. There is a guide RNA here, so this can also drive this knockout. It's an active genetic trait, if you want.
Okay. That was just while you're on that slide. No, the thing that you mentioned, though, was interesting when you were looking at the transcriptional changes in response to this expression of these antimicrobials, and you mentioned that that was on the mosquito. You haven't yet done the parasite. In the mosquito, though, do you think? Can you just explain? You mentioned it slightly, but do you think that those changes, some of them, there's a plausible pathway that those transcriptional changes could be driving the phenotype of?
Right. So I think there's many things we don't understand here, actually. And it is entirely possible that the whole effect we see is indirect. It could be that the antimicrobial peptides don't actually affect the parasite, but they're doing something to the mosquito cells, that they're in a particular nutritional state or in a particular state of stress, and that affects the parasite. I think we don't know that, but I think that is something we need to look into also, yeah.
Last question.
Okay, thank you for your presentation. I'm Claudine Mafo from Crete, Cameroon. I have a concern about the fitness course. I don't know if I get you well. I understand that transgenic mosquitoes have a reduced lifespan. If it is so, do you think that it can be beneficial, since the basic strategy is population replacement? The basic strategy for gene drive, one of the basic is population replacement. Now, if we have a reduced survival in transgenic mosquitoes, do you think that it can be beneficial for the strategy?
Yeah, I mean, it will be almost certainly impossible to make any modification that will have no effect whatsoever, I think. This effect you mentioned is only in females and specifically only in homozygote females. Beneficial, yes, of course, if females live longer, it's known that, and this is part of the model, this is integrated in the model, that also can affect transmission. This is part of the effect, is the fact that some females don't live long enough to transmit. So there could be some beneficial aspect to this particular phenotype, which is taken account in the model. But is not a suppression. This would never suppress a population, right? You could never crash a population with such an effect. Yeah.
Thank you. Thanks very much. I've got glasses in here. Thank you very much. So the next speaker, Penny Hincock, is going to give us a stroll down modeling the impacts of gene drive releases on the burden of malaria in the West African setting. Thank you. And again, I'll stand up in 20 minutes.
Yeah, within this kind of gang of mathematical modelers, consisting of Ace North, Adrian Leach, and John Mumford, who sadly passed away recently over the new year. We've been developing some simulation modeling approaches to investigate the potential of gene drive releases to control the burden of malaria in West African settings. So we're aiming to model the impacts of gene drive releases within this broad region of West Africa across a series of locations, which are shown here. So these are all square areas of 1 degree latitude, 1 degree longitude, so about 111 kilometers each way. And we chose these to span the range of variation in some key variables that we think will be influential to the impacts of gene drive releases. So firstly, variation in the prevalence of Plasmodium falciparum malaria throughout the area. Secondly, the composition of the different vector species. And thirdly, some climate variables, including long-term mean rainfall in each square and the maximum number of dry weeks in a given year. And we are looking at these QFS gene drive constructs that are being developed by Target Malaria for population suppression. So as we've seen, these spread by this homology-directed repair mechanism, whereby the construct cuts the wild-type chromosome and inserts a copy of itself during the process of meiosis, so all the offspring, or most of the offspring, end up being homozygotes. And this causes complete sterility in homozygous females, leading to suppression of the population. And despite these deleterious effects, the construct spreads from rare and can persist indefinitely in the population in theory, coming to an equilibrium frequency that depends on the fitness cost incurred by the gene drive. So we aim to investigate the spread dynamics of the gene drive in the mosquito population, as well as variation in epidemiological impacts across the different locations. So we combine an entomological model of gene drive dynamics with epidemiological model that translates the vector population suppression through to impacts on the EIR and malaria cases in humans. So just a few more specifics about the constructs targeting, again, this double-sex gene, which is essential for development in females, whereas males are unaffected by knockout of this gene. The heterozygous females experience a fitness cost, with recent large cage experiments finding that that was around about 70%. However, recent developments by target malaria are finding constructs that will have probably lower fitness costs than this. And a high rate of homing, so that there's a very high rate of non-Mendelian inheritance of the constructs. So to model the spread dynamics of this in wild mosquito populations, we use a spatial metapopulation model, whereby subpopulations of mosquitoes are connected by dispersal. The abundance of each mosquito population is regulated by the local availability of larval breeding habitat. We assume that the locations of the mosquito population are linked to human settlements, and we use these fine resolution world settlement footprint data layers to estimate the locations or the distribution of human settlements throughout the different areas of West Africa. And once we've estimated the settlement locations, we construct these polygons centered on each settlement location, which represent the catchment of the mosquito populations. So we end up with more densely connected populations in subpopulations in highly populated areas, and sparse, poorly connected populations in sparsely populated areas. And just to mention, this version of the model has a finer resolution than previous iterations, so minimum distance between subpopulations is 1 kilometer. The abundance of the subpopulations is assumed to depend on these few things. So the numbers of humans in each catchment, the availability of, or the presence of local rivers and lakes, which provide breeding habitat for the developing larvae, the seasonal variation in rainfall, and we model 3 vector species groups, the Anopheles fenestus, arabiensis, and the combination of Gambi Kalutzi, because we were unable to separate those two. We didn't have the data. But the proportions of each species group are estimated using those data layers from MAP. There is variation in the spatial connectivity across all the 16 modeled regions. The least connected areas are these Western Mali and areas in Cameroon, whereas more connected areas shown by the brighter colors with the larger circles, more urban areas. The prevalence of malaria also varies widely across the areas, which was by design. The area with the highest prevalence is this location in Côte d'Ivoire, which in 2019 had a prevalence of about 60% of malaria. So if we look at this example of the gene drive spread dynamics in this square in Western Burkina Faso, which is square 6 of our set of selected areas, we can see that the gene drive spreads, propagates to an intermediate frequency following releases. So in these animations, the red areas are low frequencies, green is middling, and blue are the relatively high frequencies. The black areas are local extinctions of the mosquito population, so these suppression gene drives show extinction recolonization dynamics, which can disrupt the spread of the alleles as the drive propagates. And on the left here, we see the population, arabiensis in this case, being increasingly suppressed as the gene drive spreads, and also showing strong seasonal fluctuations. So if we compare this to another location in Western Mali, which we can see the effect that poor low spatial connectivity has on the dynamics. So here you can see the black areas, the extinction recolonization is a more pronounced process with a broader spatial scale. And this is leading to rebounds of the mosquito population or the wild type population, so disrupting the suppression process. Okay, so as you can imagine, there's quite a lot of uncertainty in some of the parameters of these models. And in our analysis, we we incorporated uncertainty to these three parameters in our predictions of gene drive spread. Two parameters describing short and long range dispersal and the abundance of the vector population. And we did this by drawing from wide-ranging uniform distributions and rerunning the simulation several times to see the effect of that. So across all these 16 areas, we are assuming the gene drive, we're releasing a drive that has a fitness cost of about 25%. So heterozygous female fertility is about 75% of wild type. And we are assuming the gene drive can be released in either of the three species groups that I mentioned. So these plots are arranged in roughly in geographical orientation. So we can see more of the Arabiensis in green in the northern areas, and you get more of the Gambi-Kalutzi species group further south. And basically, the gene drive is causing strong suppression in most of the areas, in most of the species that it's released in, with the exception of the more poorly connected areas such as Western Mali and a bit in Cameroon. It's helpful to look at sensitivity to fitness costs because we find this consistent pattern across all the areas whereby suppression, you get very high suppression, sort of 95% across fitness costs ranging from 0 up until really high values like 70%, after which the suppression declines rapidly towards 0. And as you can see, this pattern is consistent across all the locations, again with the few exceptions of some of the more poorly connected vector species populations in some areas. So to look at the effect of that vector population suppression on malaria burdens in these areas, we use this open source model of malaria transmission dynamics called malaria simulation, being developed by the Imperial College Modeling Group. And this is an individual-based model that categorizes individual humans according to their malaria infection status, whether they have asymptomatic or clinical malaria, whether or not they receive treatment, and immunity is a function of age and time since past exposure. And the model can simulate a range of malaria control interventions, including seasonal malaria chemoprevention, vaccination, vector control with insecticides, etc. So we again assume that we consider that the gene drive release may occur in either of these three species groups. And here we have predicted impacts on malaria prevalence if we were to release only in the Gambi-Kalutzi species group. So there's not much impact in the northern areas because there's very little Gambi-Kalutzi in those regions. And further south, we get stronger impacts. So just to illustrate that, we found a 57% reduction in prevalence in Liberia, 44% reduction in the Lagos area of Nigeria, 35% reduction in Sierra Leone, Liberia. Oh, yeah. Okay, so now I'll compare if we would release in Anopheles arabiensis instead, shown here in green. This is, well, that looks a bit weird, unfortunately. But this is generally less effective than releasing in the Gambi-Kalutzi species group, as less reductions in prevalence, except for those northern areas where arabiensis is relatively abundant. If we compare that to releasing in Anopheles fenestis only in blue, again, it's generally not as effective as releasing in Gambi-Kalutzi. With a few exceptions, so it seems like this area in Western Burkina Faso that we picked has relatively large amount of fenestis, so there's a higher impact there. If we were able to release in all three vector species, this is when we get these strong impacts on malaria prevalence. And this is simply a result of the nonlinear relationship between malaria transmission and prevalence, meaning that you have to suppress transmission to very low levels before you start to get strong impacts on prevalence. So this is implying that releases in all vector species are needed to reduce prevalence down to these very low levels close to zero. However, it is likely in the initial gene drive trials at least, that releases will be in just one vector species. So we looked at combinations of interventions to see how some of the newer interventions that are being widely applied at present could improve malaria control when using combinations with gene drives. One is the RTSS vaccine, which has recently been approved for mass application in sub-Saharan Africa. The malaria simulation model has been adapted to represent the impacts of RTSS on malaria control. So we can use this model to see how well gene drive releases will perform together with this vaccination. And so here we have, assuming that gene drive are released in the Gambi-Kalutsi species group only, we have the predicted number of clinical cases averted by the vaccine in blue bars, the gene drives in the red bars, and the combination of the two in purple. And basically we can see that the gene drives are generally averting more cases than the vaccine across most areas, except for those few where, you know, Gambi-Kalutsi is not very common species. And combining both strategies is always beneficial, particularly in high transmission settings. So in Côte d'Ivoire, we're seeing around about 40% more cases averted by the combined strategy than by gene drive releases alone. Another of the newer interventions that's currently being widely applied throughout Africa are these new nets. For example, pyrethroid PBO bed nets, which achieved WHO pre-qualification status in 2018, and since then have become the predominant type of bed net used in a number of African countries. The malaria simulation model has been adapted to capture the impact of switching to pyrethroid PBO nets on malaria control. So again, we can use this model to see how well that will work in combination with gene drive releases. Yeah. And so I'm not able to show results for all the areas here because some of the simulations not finish in time. But for this subset, I think it does show the general trend in that, you know, the strategy of switching to pyrethroid PBO nets in the cyan there is generally reducing fewer cases than, averting fewer cases than gene drive releases in most areas. Combining the two strategies is again always beneficial, even though the PBO nets may be killing a few more released mosquitoes than the standard nets. And if we combine all three interventions, which is probably the most realistic case since two of them are already being widely applied, that's shown in the grey bars, that is again averting a lot more cases, particularly in the high transmission areas. For example, Côte d'Ivoire, all the interventions combined are reducing around 60%, averting around 60% more cases than gene drive releases alone. So, summary, what do our results say about where to release gene drives? Well, the good news is, in terms of impacts on vectors, we find that gene drives can achieve strong suppression of the target vector population across a wide range of parameters, describing the fitness cost of the gene drive, various ecological and environmental parameters such as landscape structure, climate processes, mosquito dispersal. The suppression gene drives can show these chasing dynamics, which are more a problem in the spatially unconnected areas where gene drives might not spread so easily. So in terms of impacts on malaria, well, we find that gene drives did perform well in reducing malaria burdens compared to some of the newer interventions that are being rolled out at the moment, such as pyrethroid PBO nets and RTSS vaccine, across most of the settings. And the gene drives acted positively in combination with these interventions in reducing clinical cases. However, it was clear that continuing transmission by non-target vectors limits the malaria control potential of gene drive releases. So thank you to all our collaborators at Imperial College, University of Oxford. Thanks to everyone from Target Malaria, and thanks for listening.
Thanks, Penny, and thank you for putting the modeling into practice. I think it's looking very positively, and it was a nice way to see it.
Thank you.
As well. Thank you. Questions?
Thank you for your great presentation. I'm new in this field, and I would like to ask a question about the long-range dispersal. So, did your model leads to how far mosquitoes can travel in terms of kilometers per day, for example? Thank you.
Well, it's a long tail dispersal, which I think on average they can travel 1 kilometer in a lifetime. That would be the average dispersal, and then there is a long range dispersal process that connects all neighboring catchments to each other. So it assumes that the mosquito has a chance of moving to any of the neighboring populations, regardless of how far away that is, during its lifetime as well. So that's the long range. That's a long range dispersal process. Yeah.
Okay, and then
Hello. Yes, please. I have a question. Your model is based on continuous releases of those gene drive infected mosquitoes.
It's just one release, actually.
Just one release.
Yeah.
And it helps to achieve 37%, or I think you even mentioned 97% suppression of anopheles.
Yeah, 95. Yeah.
And do we know the amount of release necessary to achieve that? That's 97%.
It doesn't really depend on the amount of release. If you release more, it spreads faster. But it's a drive that, you know, in theory, it spreads from a very small number of mosquitoes, so it should always spread.
And do you are releasing males or females?
Releasing males, yeah.
So once males are released, they sterilize the females.
Yeah, the gene drive, like when it's in homozygous form, the females are completely sterile, yeah.
And what about the next generation?
Yes, so it spreads through all the generations. It continues forever in theory. So it keeps spreading and spreading and spreading, potentially throughout the whole of Africa eventually, although that probably won't happen. But that's, yeah, what in theory it could do, yeah.
Okay, thanks.
okay, last question then.
Hello? Oh, yes. Yeah, I remember as if you said that the gene drive is anticipated to be less effective in Phenestas and Arabiensis. Yeah, could you know the possible factors in control to that? Thank you.
Yeah, that's really just down to the species compositions. I think it's that in most areas, there's relatively more Gambicoluzzi than Arabiensis and Phenestas. The only difference with Arabiensis could be that it's assumed to bite outdoors more, so it's a less efficient vector. But those differences, I think, are mainly due to the fact that there's more Gambicoluzzi in most areas. So suppressing that has the most impact on malaria.
Thank you. I think if there's any other questions, just for the sake of time, can you please meet up with Penny? Penny, you're going to have a very busy lunch.
Thanks.
Right? But thank you for engaging with all the questions. Okay, while Abdullah is set up, I just want to remind everybody, we're going to have a photo, a group photo taken after Abdullah's session, but I'll do another reminder afterwards. I was supposed to do it in the beginning and I forgot, so. So, a group photo after your talk. We'll wait for you. Okay, so Abdullah, also I think most of us know you. He's going to talk about field preparedness for the implementation of new vector control tools and strategies. Thank you.
Great. Thank you very much, Lisette. Good morning, everyone. I'm Abdoulaye Diabate from l'Institut de Recherche en Sciences de la Santé, Burkina Faso. And I'm really grateful to Antivec, for the invitation. I must say that Tony, Nikki, and others basically had made my life easy by setting up the scene. It's clear from what we heard and saw that gene drive holds a lot of potential for Africa, but at this point in time, it's still a piece of DNA in the vial. So how do we take it from there to the field where it can impact the lives of those who need it the most? So this might sound like a little bit cliche, but I always thought that it's very important for us, to start the talk with this. So to remind people why we are doing this. Mostly those who are opposed to the technology that we are developing. We are not doing it for the sake of science. We're doing it really because we want to save life. As you can see, malaria is a real killer. 200 million cases worldwide, that end up sadly every year with about 500,000 deaths. And this is not a random statistic on the piece of paper. Behind each of these 500,000 deaths, you have a personal tragic story of people. So mosquitoes, malaria mosquito principally, have evolved to escape most intervention that we have today. And it's clear that when we look at the general consensus that we have today, people think that if we don't have additional tool to complement the existing one, we may never be able to cross the last mile of malaria elimination, no matter how much money and effort we are putting in there. So it's really clear that we need to innovate. So, innovation comes out of necessity. It's a mean to an end. At this point in time, in our fight against the disease, I think, especially in high burden country like Burkina Faso, it's extremely important for us, to have bold idea that can have a really very big impact. Good so. If you look, for example, at the pipeline of the research that is out there now, we have so many good ideas that are there. Anything, from the new net, the ATSB, spatial repellent, genetically modified mosquitoes, Wolbachia. We could not be in a better time now. But sadly, what happened is that if you look at the pathway from the discovery to the public market, you will find that basically it's full of pitfalls. Most of these ideas will shine once, but they will never make it, to the heavy hand of the gate. It's going to be really crushed in the pipeline. So obviously, we need to do something about that because the complication really is science-based, regulation, risk, and the public acceptance. To illustrate my case, for example, it's take, for example, to screen 5 million compounds to end up only with 3 molecules of insecticide. That's a lot of money and time for anyone to invest. And even so, sometimes even when you discover a specific product, the pathway, from the discovery to the market is not really the shortest one. What happened, for example, if you take permethrin, it took permethrin almost 30 years after it has been discovered in 1933, to get to the public market. And the reason is that the public health market is really quite small, but beyond that also you have problem of regulation. So here again, I'm showing you, some of the challenges that we are facing in this space of vector control and developing, the new tools. So I'm just going to take them one by one and walk you through. so that you can understand the challenges that we have and how we need, to get ready for that. So what is going to be said basically is going to be relevant to all the new incoming tools. But I'm just mostly going to focus, on target malaria for gene drive. This is a kind, of illustration of most of the challenges and opportunities that we are facing. So Target Malaria is a not-for-profit research consortium made of scientists of different backgrounds all over the world, but mostly in Africa. We work in Burkina Faso, in Mali, in Uganda, Ghana, and then in Cabo Verde. Our ultimate objective, in fact, is to develop and share with the malaria community a genetic vector control tool that can deplete mosquito population and ultimately halt malaria transmission. You will say, that's really fantastic. The only problem, gene drive has never been tested anywhere in Africa. So while the technology brings a lot of hope, it also carries a share of fear and skepticism. So one thing extremely important to keep in mind, the safety issue, as it has been really very elegantly coined by Mark Benedict. He said safety deficiency is the one aspect that might not be easily remedied. One can gather more baseline data, lobby for political support, and build capacity. But if we cannot provide a plan that people consider safe, the effort is really dead. So for this reason and others, basically, Target at Malaria has decided, to adopt what we'll call an incremental phases, whereby we will go, with the sterile male self-limiting construct all the way, to the self-sustaining male bias. But each of these constructs will go through a stringent pipeline of testing, as you can see here below. So where we will test, in London in small cages, all the way to the small scale field release in the field. That's really fantastic, but that's a conceptual framework. Question now, how does it translate really in the field? So when it's come, to testing technology like gene drive in Africa, I think it's rely mostly on 4, if not 5 pillars. So the first one is the technical competencies of the human capital, along with the infrastructure that are required, to conduct good science. Target Malaria has built an ACL2 insectary in each of its main sites to host, the transgenic line, and every single aspect of this insectary has been looked at, from the design of the insectary, the biosafety manual, the SOP, equipment and maintenance, all the way to the waste stream. And all the personnel that are involved in this project went through a really very stringent customized training. And at the end of this training, basically, we have a certificate that we can put in the personnel file for, future verification by the National Agency of Biosecurity. And here in this picture, I'm just showing, some of the training that we got in Italy, we got in the US, we got also in London. That was from my lab and also the lab from Amadou Koulibaly in Mali. So, excuse me, in 2015, before even we get, a single transgenic line, we committed an internal audit of the insectary. So that was followed by a second one, formal one, by the National Agency of Biosecurity in 2016. Then a second one in 2022 when we refurbished, the insectary. Factors were measured against the general lab practices, the specific target malaria policies, the national and international requirement containment facility. And I can tell you that the National Agency of Biosecurity was really very satisfied with the compliance. And at the end of this audit, they provided us, with what I will say a certificate of compliance and conformity. And that allow us, in fact, to import genetically modified mosquitoes. And we import them twice, the sterile male and then the PMB. And I'm just showing here the process of transportation from Italy to Burkina in Ouaga, where our personnel will go and then, collect the samples. And as you can see, the lab manager basically is inspecting, some of the eggs on arrival, and then just also seeing, the quality of the eggs. And then when these eggs were dumped into water, and then they hatched, I can't tell you how happy really we were. So as you can see, these mosquitoes are now sitting in our lab. So they are going through a series of testing that we are doing. And hopefully at some point we will be able, to take them to the field where we can do a small scale field release. So the second pillar, basically, this slide is from Frederick sitting here. He was helping us, in Target Malaria. So it's a technical scientific data that really need to be collected, to predict the utility, of gene drive in the field. And as such, I must admit that, Target Malaria is conducting extensive entomological field activity in most of its field sites. And data that are collected basically are going to feed into the insectary data for the modeling, the ethics, the regulation submission, are obviously also for the modeling. And obviously also we are diving really very deep into the population, genetic and genomic, to understand, the extent and magnitude of gene flow between and within population. So, nonetheless, even though that we are doing that, it's absolutely clear there are still astounding number of questions really that we need to address before we are able, to take these mosquitoes to the field. And some of these questions are, written down here. I'm not just going to read them for the sake of time, but it's really very important to notice that we got, a fund from Welcome to Us recently, to tackle this question, and we've set up a really very good consortium with some of the best brain across Africa. Charles Wonji is out there somewhere, sitting behind, exactly, so it's part of the consortium. So it's a really very vibrant consortium, and then we will be looking at the population ecology, genomic and genetics, and also modeling. Now, the third pillar basically is the stakeholder engagement. If there is really one aspect that you cannot afford or have the luxury to miss, it's the stakeholder engagement. Because when it's come to gene drive, the release of gene drive in the field, trust and full transparency are fundamental pieces of our work. I cannot just pop up in a village with a bucket of mosquitoes on the assumption that I'm a scientist working for the good of the public. It will not take anyone, a PhD really, to understand that I'm respectful of their value or not. So it's really extremely important, to get it right and then to engage the community. Then the question is, who is our community? Burkina Faso count about 20 million people. So potentially all these 20 million people are our community. And we need to find a way, to communicate with them. So it's clear that you cannot talk about gene drive to your Minister of Research the same way you're going to do it with a random passenger, at the bus station or even the imam of the mosque. So you need to come up with a strategy, develop key messages, and then be able, to deliver them really very effectively. So at the end of the exercise, what we did is that we came up with a pyramidal structure of communication and engagement. So where we started at the bottom of the pyramid, with the villages, where we did the communication, all the way, to the highest level of the pyramid with the highest authority in the country. And in between now, you have different layers of the society. Then the question is, how do you even communicate on something that you don't have yet? So what we try to do is that we did our communication step-by-step approach and inclusive engagement, and also we are doing, the research in full transparency. So I'm just showing you here a kind of snapshot of stakeholders that have been engaged in Burkina Faso, but it's way, way, way beyond that. This is just a kind of snapshot. So we engage, a lot of people, that the traditional authorities, the religious authority, we went to the parliament several times. All the key ministers have been also engaged, our peer scientists, all the municipality, the civil society, the media, even at the village level, where we came out with different media, way channel of, communicating with them. So the fourth pillar basically is the regulation. So when you take this kind of product, where, the question of boundaries is going to be problematic between countries, so it's really extremely important to ask yourself in the first place, how is it going to be regulated? Who is going to do it? What legislation is needed for that? And what about the rest of the country? So if you take specifically Burkina Faso, we have already, a legislation which is in place that allow us, to deal with this. So we have the National Agence Nationale de Biosécurité, which is already dealing with that. And then this has been functional since 2020, since 2005. But the real question is that, what about beyond Burkina? Because the rest of Africa is one continent, but you have 55 countries. So it's clear that it's going to be extremely difficult, for the technology developer to run in each of these countries, to look for regulation and then permission to be able to do the work. So someone has to do it, and it has to be fixed. And this is something which is done by the AUDA NIPAD, which is a technical branch of the African Union. So the way they see Africa is not like 55 countries. They're just looking at Africa as a regional bloc. So for example, in West Africa, you have the ECOWAS, which pulled together 15 countries. So the question is, hey, all the regulators in all aspects in these areas that are there, question is, something really very exciting is coming. How can we work together to make sure really that we can harmonize our regulatory system in a way that the scientists working on this don't have, to run in all the places to have permission to do the work, so that if they submit the dossier, it can be approved, at the regional level. And so they're taking example, from Burkina to replicate this model in other part of Africa. And so they will do the same thing in East Africa, Central Africa, and on South Africa also. Now, I'm gonna finish my talk with the capacity building. I used to think that there is no spontaneous generation of good scientists and leaders. We all are the product of the education system we went through, and that has a long-standing experience on who we are from the primary school to the university. And that's really very important. And sadly, if you look at Africa, we have one of the weakest research and science and technology institution. And unfortunately, Africa bears the highest burden of most of the preventable diseases. But we have the lowest technology development, and so our impact on the global research is really very weak. It's clear that all these tools that we are talking about today are real potential for Africa, but it will not take us anywhere if we Africans are enabled, to run it on our own. And so far, it's really extremely important for us to set up a kind of capacity building program that will help us, to lift up, all these programs to take it to the next level. And this is exactly what we are doing with PAMCA. So we have a specific program now where we try, to reinforce the capacity of people at the National Malaria Control Program level, at the operational level. And so we working on specific aspects, the advanced molecular biology, population genetic and ecology, modeling, trial surveillance, and monitoring. And this activity is being done now in the 3 centers of excellence of PAMCA in Africa, which is in Burkina Faso, in Cameroon, and also in Tanzania. We are also, conducting short training courses on gene drive with PAMCA, and this has been done since 2017. Now we are in the 5th generation, where we, teach people, we raise awareness about the gene drive, but also, we impart them, some of the knowledge on gene drive. And so far, so we have trained about 134 people, and this has been done more than 30 countries by now. And this is very quickly the list of the facilitators that you will recognize some of them here in the room. And also, since 2019, we've set up a Center of Excellence funded by the World Bank, where, we're tapping in the brain of the collective student, across the different universities, to build the next generation of scientists. And this has been done since 2019, as already mentioned. So we have recruited students all over Africa. So they're coming to Burkina, having the training, and then taking back the knowledge to their home institution where they can apply it, and then they're going to be at the decision level one day also, so they will be aware of what is gene drive, and if they have to take a specific decision, that they can do it. And finally also, so we got recently, a grant from the BMGF, Bill and Melinda Gates Foundation, collaboration obviously with LSTM. Tony is sitting here, where the objective in fact is to build a really, very good capacity building program where we can, build the next generation of African scientists and make sure that they have access to the cutting edge equipment, but also coached, by the leaders in the world, so that we can have, generation-driven, technical generation-driven human capital. So now I'm gonna finish with the plan for development, deployments, sorry. So if you look at most of the technology that we are developing, so the list is really very important here. Anything for the biocontrol agent, the paratransgenesis, Wolbachia, genetic modified mosquitoes. The real question is, when we get to the point where we have a final product, how are we going to scale it up? Who is going to do it? And really very importantly also, who is going to pay for it? This is something, these are really completely open questions. We don't have answer yet, but it's really extremely important for us, to start thinking about it, so that by the time we have the technology ready to go to the field, to be deployed at large scale, we know really how to proceed with it. And so, in my mind, there is absolutely no doubt that this has to be a public-private partnership. For the simple reason that, the public sector, really the machinery is really very heavy to lift. That's one thing. But the second thing also is really grounded in bureaucracy, and it's going to have a very bad impact, on the implementation of this if it's only based on the public. And not even to mention that the quality really that we want may not be there. So it's extremely important for us, to start thinking about that and see how we can pair this to have this kind of collaboration, so it can help us, to take the technology to the next level. So the last slide here basically is that we have been working, on some of these, But this is just one of the products that we have been working on, which is biocontrol fungi, which is metarhizium, that we have been working on for really quite a while. So we have not got to the point yet where we have a specific product ready to go to the field. Nevertheless, we have started, discussion with some collaborators in China to see how they can help in the future to set up, a mass production facility in Africa so that we are able, to produce this our own and then be able, to implement it at large scale. So obviously, just for your record, we got awarded, the Newcomb Cleveland Prize for this seminal work, and this was in 2020. This is the reception ceremony in Seattle, and as you can see, even Usain Bolt could not do better than. So thank you very much for your attention. Must say that I needed, to have a lot of names here. I have plenty of students in the room. They are all contributing. I have a lot of collaborators, but it was really very difficult for me, to put everything here. So I just put thank you for everyone, to everyone, but they will recognize themselves. Thank you.
Thanks very much, and thank you for sharing your passion with us and for asking some serious questions that needs to be out there. Questions?
Thank you very much for that very brilliant presentation. I just want to shift you a little bit from the health issue to the agriculture and come back. Most of what you presented has been done in the area of agriculture, for example. I remember in Burkina Faso, there was the introduction of BT cotton, that the thing you remember very well was a genetically modified crops. Yes. And you know the story. Also, the entomopathogen, the biological control. What I'm trying to say is how much, as you go deep in this type of technology, you have shifted to learn from other aspects like agriculture, that similar technology are being done, and what are being the mismatch, the failure, the success that we can learn from and accelerate this type of deployment. Thank you.
Great. Thank you very much for the question. It's a really very important one, as you really mentioned it. In fact, so metarhizium or genetically modified mosquitoes were not, the first genetically modified organism to get in country in Burkina Faso. So we had, the genetically modified cotton a while ago. Well, but the big difference that we have to see that one is agriculture and then the other one basically is your, health. I think that one of the big issues that we have, I don't think that's only in Burkina Faso, that if you look across, and then so many people that we have been talking to, is that very often people look at, the agricultural side of the thing as a business. So they feel, well, okay, they're coming with genetically modified organism. So what is going to happen to our seed? Are we going to lose them forever? And then are we going to depend on them? That's the first thing. And then the second thing also is that, well, okay, these people are coming, to do business on our back. So should we allow that? Absolutely not. But when we shift, to, well, I didn't do the genetically modified cotton, but when, we started working, on genetically modified mosquitoes, I mean, the dynamics is really completely different. As I said again, once look at the conventional tool that we have in hand today, we all aware that these tools are not enough to take us, to malaria elimination. So at the National Malaria Control Program level, and then the Ministry of Health in Burkina Faso, and even beyond, so many stakeholders that we have been talking to, they really recognize the added value of the technology. Because one thing is that they feel like, okay, technology is really very important if it adds something to my life. If it doesn't have anything to my life, so sometimes I will be really very careful about it. So, but people really see the need here. And then I wouldn't say that it has been easy, because even those we having some struggle in the field, but it has been a little bit more accepted than, the genetically modified cotton. That being said, now, at the regulatory side, basically Burkina has been, one of the first countries, to have the regulatory system in place. And this has been put in place thanks, to the introduction of the cotton BT. So they have learned a lot from this process, and then they try, just to translate that to what we are doing now. At the science level, so I will say that some of my colleagues, we all belong, to the CNRST, because they are from INERA. So they have been working on this genetically modified cotton for quite a while. So we are discussing a lot. So we're trying to understand from them also what have been the failure, and how can we do better just for us, not to face the same challenges that they have faced in the past. So I must say, so far, so good.
More questions?
Thank you very much and nice presentation. You've been talking a lot of the communication with the population, but how will you be talking of a communication on something that you are not yet sure that you will establish even in Africa?
Okay, so Madame Paris is here, and then Barry also, they will have, presentation on that, specifically something really is going to be focusing on stakeholder engagement. So they will go in depth, but anyway, it's clear that we started, the project really very long time ago. It's like 2012 in Burkina Faso, and then in 2023 now. We don't even have the project, the product yet. But one thing is really very extremely important is that you need to keep in mind, that everybody really need to keep in mind, is that this technology is not just about science. Science is really fun. It's a lot of work. It's very smart. But one of the critical elements really is how do you really make sure that you get the social license from your people, to do the work. And you can only get that if you can build trust with them. And then you start working so that they have also to understand the process of what is research. Research doesn't mean that I'm working on that now and then tomorrow I'm just giving, a specific product. It's a long process. So we need, to test. And since 2012, we've been working with the community. So this has been really clearly explained to them. They very often, come to our research institute. They see, the mosquito that we have there in the insectary. They ask questions, and then they know that it's going to be a long process. So they're working alongside with us just to make sure that we can get to the product.
Thank you. Any other questions?
Thanks for the great work, dear Bate. A quick one, especially on the gene drive trainings. What has been the impact over time? I think you've said that you've done about 5 of them. Do we have like a way to follow up to see what the effects that has had, other than just doing trainings and then maybe people, the fact that the technology is not with us here in Africa yet, how do we measure the impact? But also, how do we intend maybe to have country-based, so that that training, instead of it being done like once a year, but also even countries adapting similar trainings, so that you increase the pool of people who have the technical know-how?
Yeah. So one of the clear things that we can see already is that with this training, so we're raising awareness. Because I can tell you, the early beginning of target malaria, if I will go, to a scientific meeting somewhere where I have to present, I will even hide. I don't want people, to know that I'm from target malaria. Because, at that time, people didn't have any information about genetic modification. So there was a lot of misconception. So we really took time, to talk to people, to explain things. And then you have different categories of people. Some people really, they're in doubt, but they want to have the right information and make their own decision. But so what is really very important, so you have to make in a way that the space of communication is not filled up, just by the anti, because you have a lot of false rumor there. So you need to make sure that you are close to these people, and then you can give the communication. And so a lot of people coming to us, doing the training, they're taking it back to their home institution, and then they're talking, to either the Minister of Research level or at the Minister of Health level as well. But one of the things also that people are asking, well, okay, this is really good. Those who want to go a little bit further, they want to understand if beyond the short training courses, because this is just 3 days, can we give them, experience on hand-on experience, training, so that where they can come for at least 2 weeks, they sit with us so that they can go through the theoretical thing and then also do some practical. And this is something that we are preparing. Hopefully, this year or next year, we will be able, to deliver that. So I was talking to Charles Wanji. Probably the first one is going to be in Cameroon, maybe this year or maybe next year, and then we'll see. I'm not sure what was the second question. Yeah, well, okay. So as you know, you part of PAMCA already anyway. So we have 3 centers of excellence of PAMCA already. So the idea is that, okay, we doing it on the yearly basis. So how do we also make sure that this knowledge is embedded at the country level and making sure that at least starting, with the Center of Excellence of PAMCA so that they can deliver these courses themselves. And then from there, we can see how we can expand it, to other countries. But it's coming really, very nicely, I must admit.
Thank you. Sorry, we're running over time. So if there's any questions, please meet up with Abdullah during lunch. So we're going to do the photos. Michelle is here. We're going to do a group photo somewhere. Okay. Yes. Okay, thank you.
one folder.
Yes, I just want to copy some over.
just use this one. Okay. Pray silence. Never worked. Thank you. Okay. Okay, I'm the chair for this session, and the first speaker is Dr. Lea Padetoy from IRSS at Burkina Faso, who's going to talk about their efforts on stakeholder engagement around genetic control in Burkina Faso and more widely.
Thank you. My name is Leah. I'm a social scientist from Burkina Faso, and I'm leading stakeholder engagement for target malaria also. I'm going to talk about the productive collaboration between scientists and non-scientists, a requirement to improve the ownership of gene-derived technology. When we are talking about the gene drive, we are talking about a science. And science is one of the humankind's greatest initiatives to improve human well-being. But that does not prevent it from some challenges. For instance, the skepticism over the funding. And we have a lot of examples on that. Why the scientists work hard, for instance, to come up with the COVID-19 vaccine, we got some protests against this vaccine. We have the example also for GMO crop, GMO mosquitoes. You know, the effectiveness of technology does not guarantee the adoption. When most of the time, we have the technology and we spend long-term delay between the availability of this technology and the use by the end user. For instance, for the bed net, between the availability of this tool and the adoption, we spent almost 10 years. In agriculture, we have also a lot of examples. When the scientists came with the efficacy technology, but the end user don't use it. If we want to improve the ownership of gene drive, we need to anticipate on those challenges. For that, we need to redefine the relationship between science and end user. You know, the concept of the relationship between scientists and end user is an old debate. We have a lot of concepts like the community participation, with the concept like public information, public education. But the problem is, for most of this collaboration, it's about one-way communication. We need to go beyond this one-way communication by establishing the two-way dialogue with the concept like the knowledge and experience sharing, the concept like co-development, co-working, co-thinking, co-construction. But to co-develop, co-think, there are a lot of challenges. How we can do this? And practically, we can say that in terms of process, one of the first things is about the mindset change. The mindset change from scientists first. Scientists should recognize that they don't have a monopoly of the knowledge. And the non-scientists, they have their own knowledge roots in the social, cultural, professional, political landscape. and that can impact negatively the adoption of our funding. We can provide the room for the stakeholder to impact our research process. From the non-scientist perspective, they need to change their mindset, to trust to the scientists, to understand that the development of the technology, the development of any scientific thing comprise some uncertainty, the deadline. And the non-scientists should be ready to take part, to play an active role in the development of the technology. But if we take a technology like a gene drive, if we want to have this productive collaboration, the first challenge is among scientists itself. You know, there are a lot of and diverse expertise working around gene drive. We have the expert in biology, the expert in medical anthropology, in genetic, social science, expert in risk. And all of those expertise, they have their science, have the specificity, and there are a lot of differences. How all of them can establish a dialogue to be sure that each other can understand what the others say. When I'm working on the biology, how I can understand the language of risk, the regulatory. We are working on the same thing. If we want to collaborate, the first thing is to have the same, almost understanding the meaning of the thing. This is one of the big challenges. Another is, when we are talking about the productive collaboration, we are talking about the collaboration between the scientists and non-scientists. We recognize gene drive is a complex topic, even for scientists working in non-genetic area. Above, stakeholder who lack formal scientific training. How to talk about chromosome, how to talk about DNA, to be sure that we can understand each other and we can collaborate. But in target malaria, we have some example when we start this collaboration, and we notice that stakeholder, they can understand DNA, they can understand chromosome. Another challenge is about the diversity of the stakeholders. We have the stakeholder that we can call the affected community, meaning that those living in the area where we do, for instance, the release. We have the decision makers. We have the regulatory. When we are talking about the collaboration, where should we start this collaboration? Which category of stakeholders? We need to prioritize and find the appropriate way to do this collaboration. It's difficult as a scientist to do that, but we can do that. And when we do that, we will get a lot of advantage. For instance, we will empower stakeholders to get the informed decision about our gene drive technology. Stakeholders could impact the research process. And because of this productive collaboration, gene drive technology will become something honed by scientists and non-scientists and treated accordingly. I'm going to share with you one of our example in target malaria around this productive collaboration. You know, we are working now on the, as a scientist, we are working on this gene drive technology in the lab, but we plan to take it on the field for the release. When this time comes, we will need the public acceptance for this release. How will we get this acceptance? Which model, the agreement model, we will use for this release? In target malaria, as you know, we start with this serial mail. And since 2019, the date for the release, we start this co-building, co-construction of the agreement model with the community, the affected community. And those model has been supported by a lot of social science to analyze the power structure in the community, the decision-making process, the legitimacy issue. Who can talk on behalf of who? We work with the community to co-develop the design of the model, the selection of community representative, the validation of the committee, the design of the acceptance form, as well as the traceability. And now we are working, again, we are continuing to work on this model for our second step with fertile mosquitoes. And we taking the lesson learned for our previous model, we now export the discussion with the external stakeholders, and we are working on the concept define the community, because when we release, who will be considered as a community? Just the village where we do the release, or at which level we can talk about the community? And in this model, we have a strong components for the country, because as you know, target malaria is working in different countries, but at country level, the stakeholder has involved strongly in this model to be sure that when we get at this point, we can have something who can be acceptable by the stakeholders. Thank you. No question. No.
Thank you very much for your presentation. I wanted to ask a question in two steps. The first one is the breakdown of, you said common concept, or how do you break down this complicated chromosome and DNA stuff when you're talking to non-scientists or maybe the media? And secondly, how do you deal with reputational damage and misinformation in the context of innovative technology like gene drive? Thank you.
Thank you. You know, when you are thinking about DNA, chromosome, yes, if you are come to the village and start to talk about this, by using this word, it becomes difficult. But beyond this word of chromosome, there is explanation. And this explanation makes sense in the community, because, for instance, if you want to transplant that, if you are married with someone and you have the baby, and the baby will take something from the father and the mother. You know, this kind of thing, it makes sense. They are used. That means we use the explanation. We don't use the word, but the explanation. And based on this explanation, the community come with the local concept to express this reality. This is for the first question. The second, about the anti-GMO or the protest, sorry? Yes. For instance, in Burkina, we got this also when we did the first release. But, you know, because of this dialogue, two-way dialogue, we build the trust. We allow a stakeholder to see what we are doing in the lab. They reach a certain level of the understanding of this science. So when some people come and try to disturb, try to give the misinformation, people at the village correct this information by saying that we know, we understand what they are doing. What you are saying, it's not true. That's why establish that two-way dialogue. Take time to talk with people, show them things, explain them. They can understand and support. And they can feedback also on the research process and improve it. Thank you.
Leah, I have a question. So at the time of the release, there was some demonstration in Ouagadougou. And how do you deal with a demonstration that happens in the country of release, but very far away from the actual release site? What was your strategy there?
Our stakeholder engagement strategy is inclusiveness, meaning that we are working on the western part of the country, but we engage across the country. We engage the different category of stakeholders. And when those protests came, we got, I can say, some response from the stakeholder itself by saying that, okay, even the stakeholder at Ouagadougou, which is the capital, because we engage them, because they are aware of this project, the response to this, all of this, noise on the research. It's strongly related to the strategy of stakeholder that we put in place for this release.
Okay, thank you. Any more questions? Okay. Next up, we have Nuru Bari, who's going to talk about operationalizing stakeholder engagement for gene drive research in malaria elimination in Africa, and how to translate his and her guidance into practice.
Okay, good afternoon. I understand that it's not easy to talk just after here your bosses. So it's really stressful. Uh, so thank you for giving me the floor, Tony. I'm Nourou Barif, an anthropologist from Institut de Recherche de la Santé, Burkina Faso. Through my presentation, I would like to share and discuss later on how we as the stakeholder engagement teams for target malaria, we were able to operationalize the stakeholder engagement for gene-drive research in malaria elimination in Africa by translating the guidances into practice. On this first slide, I would like to highlight a couple of realities that we are living on. The first one is, to mention the fact that here in this warming room, we all, hope, we have a great hope on the fact that gene drive is seen as a potential transformational tool for malaria elimination. That's the first fact. And the second one is to also mention that the international guidances and also here in this room, we strongly think that stakeholder engagement is one of the components that will strengthen that technology in development. But unfortunately, there is actually very few, practical recommendation on how to proceed, how to really achieve this effective engagement, how to how to engage, and also how which modality to use. And this is why Target Malaria, as a pioneer research consortium on this technology, has been reflected on how to navigate between those international guidances and also by taking into account the specific context on which the technology is operating. And this is why the rationale behind this specific communication I'm giving right now, it is an open access paper which already has been published on Malaria Journal. Target Malaria, through its vision and missions, has already developed a couple of values that we think that we will be helpful for us to strengthen the stakeholder engagement. And those values are the values based on the excellence and also the co-development, which is an important component for the stakeholder engagement. But we are talking about stakeholder engagement. What is the stakeholder engagement, really? So we took this definition from the IFC, which defines the stakeholder engagement as about building and maintaining constructive relationships over time. It is an ongoing process between an organization and its project stakeholders that extends throughout the life of the project and encompasses a range of activities and approaches, from the information sharing and consultation and through the partnership by taking into account the participation and also the negotiation. But how we really and practically were able, as a stakeholder engagement team in Target Malaria, were able to really navigate between the international guidances and the context specific realities. First of all, we, at the beginning of Target Malaria, the Target Malaria activities in Burkina Faso, as Professor Jabate just mentioned here, in 2012, there is very few, guidance on how to proceed on the stakeholder engagement. So for the stakeholder engagement for Target Malaria, the stakeholder engagement is really very requested as something to help, going on the process. And by doing so, we were able to, the focus were to understand better, the local context and also the social dynamics, and also having a good understanding of the socio-political landscape and also the regulatory as Professor Jabate mentioned. And also, we were able to elaborate, the principle for engagement by focusing this principle on the component that I just mentioned before, the co-development as a pillar, by taking into the advantage from the international guidance and to improve the process of engagement, but also by reinforcing the context-specific approach with the international guidance. So this is shown in this figure, but as I can see that the colors are really not easy to read. So finally, the last thing that I have to mention is how we were able to really translate the guidance into practice. The documentation that we had in the the international guidance mentioned the adaptation to stakeholder preferences. The NASEM document and also the WHO document all mentioned this recommendation, the need to better have a better knowledge on the stakeholder values. By doing so, we were able to put together how the social ethnographic studies which help the team to really have this good knowledge. And also, one of the recommendations is rooted in the principle of co-development, and as a practically we were able to really adapt our research plan to the stakeholder feedback by taking those feedback into account. But we were facing a kind of challenges due to the time issue, and the research is taking long time, but we were able to really manage this time by confirming and really interacting a lot with those population, those stakeholders. And the solution were to establish the two-way dialogue that's exactly mentioned by Madame Paré, and find the right balance between our needs as a researcher and the community or the stakeholder needs. And the final one is the inclusiveness. Tony has just asked one question, and the inclusiveness is really important in the process, in the strategy of stakeholder engagement, because the international guidances recommended these important recommendations, and we were able to promote the participation of all categories of stakeholder by including all together the stakeholder, including the one who really affected and the vulnerable population. But the challenge were to which level of implication for the various categories of stakeholder, which level is really the best one that we can say this level is reached and we can stop there. And something not necessarily culturally accepted and appropriate on some fields, but we were able to establish the co-development as an alternative for doing so. Finally, there is several recommendation guidance documents raised. Those recommendations, the existing recommendation raise tension because they're leading the misunderstanding of communities or local policymaker, and they're sometimes culturally not inappropriate. But the solution is maybe to the urgent that the researcher have to open more studies, to enhance the better implementation of the international guidance by taking into account the context-specific realities. Thank you so much for your attention. And
question? Okay, I have one, Barry.
Yeah, okay.
It's actually the last thing you said there. You were talking about which level. I take that to mean, how do you know when you have done enough stakeholder engagement? It seems tricky to me to know when you've done sufficient engagement and when do you stop.
So thank you, Tony, for the question. The problem with the stakeholder engagement is the fact that it's really is ongoing. It's continue. You have continuously have to do the the engagement. So we don't really have the right time to say that that time we really mention or achieve the stakeholder engagement. So it's ongoing, but the fact is we can have the strats when we see that all the stakeholders are acceptable, they accept our research, they accept to co-develop the research together. We can say that we really reach the goal.
Yeah. Okay, quickly, and then afterwards, Charles. Charles behind you has a question, so please be brief.
One of the way to know the how you reach this level, it's the understanding. When you are working on maybe the phase one and the people can understand the research, this is also one of the measure. Thank you.
My question regards the regional interactions, because Burkina is part of West Africa, I think ECOWAS. You are engaging with stakeholders in Burkina, but do you think it will be enough to start any action, any release, knowing that you have neighbors who may have a say also, because mosquitoes, they don't know the national boundaries? So in relation to that, how do you see things happening in the future for this really to happen?
Okay, thank you for the question. In our strategy, in our stakeholder engagement strategy, we start really to work in Burkina Faso at whole, but we also included in our strategy, the fact that we have to also engage beyond Burkina Faso. So the team has already started talking on some of countries neighbor to Burkina Faso, and it's ongoing, and maybe Madame Pare will. Okay. I don't know if I answered the question.
You can answer it more fully offline because
Okay.
we have to move on.
Super.
Okay, so next we have a late entry, a naughty boy who didn't send his homework in to Michelle on time, who's going to talk about the African Genetic Biocontrol Consortium. He's already handing out flyers, of which I have one. So this is Willy Tanui.
Thank you. I may not be as naughty as Tony has mentioned. I think he's the one who is naughty because he's adopted my name. Instead of using Tonui, he's using Tony. So that's how naughty he can be. So my name is Willy Tonui. I'm from Nairobi. I work as a consultant. I started my journey of life as a scientist, and then I became a regulator, and now I'm doing international consulting on issues of gene drive. So this afternoon, I'm going to talk to you about addressing some of the knowledge gaps that I've had from you guys since morning, and especially Professor Diapate, when you enumerated about the challenges that they has been facing in their realm of work. So I don't know, from this thing is not moving. How many of you think that in the next 10 years we'll have a gene drive field trial in Africa? Only Nikolai. How many? What will it take for us to have it in the next 10 years? Using a non-GM gene drive product that they have been testing, they've taken more than 10 years. So what will it take using public participation that they have talked about to try now and explain that the trial that I'm using now has a genetically modified organism? So think about that. So we do have our African Genetic Biocontrol Consortium that is trying to address some of the challenges that we go through in terms of genetic biocontrol studies as well as gene drives. And you are aware that we have countries and we also have regional bodies like the African Union. And there was a need for a group of experts now who are from the non-governmental organizations to play a big role also in terms of trying to explain, to support gene drive research and development. So we came up with member organizations in Africa, and we decided to establish a consortium. And this consortium basically is to enable members within the consortium to work together or to work individually. And we did this so that we can attract funding as well as informing our stakeholders. So far we do have this consortium launched in November 2020. And we do have membership that include the African One Health Network. These are a body that's recognized for spearheading One Health Network in Africa. Then we have the African Biological Safety Association, regional body also that has a mandate of looking into issues of biosafety and biosecurity in the African continent. Multilateral initiative that you know about WHO and initiatives that are associated to MIM. Then we have the Network of African Science Academies. This is a body that is dealing with academicians. Then we have PAMCA, and then Gene Convene Global Collaborative. We do have a website, and I want to let you know that the website is translated into French, Portuguese, Arabic, and also Swahili. And then we do have newsletters that we transmit on regular basis, weekly and also monthly. So I want to let you know a bit about Gene Convene Global Collaborative, which I'm also associated with. The role of Gene Convene is to advance best practices. And we do have a website there. We offer technical information, advice, training and coordination for research on gene drives and genetic biocontrol technologies in general. And we do have a website. And one of the strengths of the Gene Convene is that we do have what we call Gene Convene portal, whereby we do have, it's a virtual institute where we do have information on genetic biocontrol. And we do offer this information through the virtual institute. So frequent questions about gene drive and periodic information that comes out from time to time on genetic biocontrol activities. So within the consortium also, we do have the training and capacity, whereby we do provide regular in-depth discussions, through webinars or face-to-face meetings, through workshops. And then also we have a thematic area on research and development, where we do support now country-specific development and expertise. And so this, we hope that it will promote a consistent approach to research and development in the region, and also help in delivery and also development of best practice documentation. And also issues of One Health are growing. When you heard about Cartagena Protocol recently, one of the decisions was about One Health issues. And so One Health and gene drive and related issues are coming up very strongly within the Cartagena Protocol. Then we have vector control and conservation. This is spearheaded by PAMCA. So within this thematic area, we hope to support training now and development. And PAMCA has been doing very well. And as a consortium, we provide a platform for disseminating and addressing this information to the public. I know the questions of transboundary movements are very, very, very critical, as Charles Wonji asked. And these are some of the issues that we try and address cumulatively through all these member organizations that we're working with. So we have a social media presence. We do have Twitter, and we are mainly on Twitter. And whenever there is any hot issue about genetic biocontrol, we do. So please, we hope you like us. Then we also have monthly newsletters. And so far, we have developed very many newsletters, at least monthly basis, Last year, we produced over 12 of my monthly newsletters, and this has content that related to genetic biocontrol. So we would like to have as many people joining us. So you can send to us an email and to join our mailing list. So if you join our mailing list, you'll be able to receive our weekly newsletter, which you call Gene Bio Weekly. And this Gene Bio Weekly gives you information about what happens in the world, what are new things that are people talking about, new publications, and also sharing opportunities for training. So unless you join the mailing list, you'll not be able to get this mailing list. So please send to us an information. And we can also, we have also this Gene Bio Weekly is translated into French. So in the past, we have had issues with French speakers, but we now have a direct translation of in a French fashion. Last year we did a training on risk assessment. So as a consortium, we have recognized that the interface between the regulatory agencies as well as the scientists and institutions is very is not there. The gap is still huge, and as a consortium, we are striving to bring the regulators together and the scientists. So one way of bringing the scientists is to enable the institutions to have capacity. One of the requirements for approval of a genetically a project involving GMOs or genetically modification, you need an institutional biosafety committee. So, and the institutional biosafety committees are the ones who talk to you. They talk on behalf of the scientists to the regulator. So it's important that the institutional biosafety committees are strengthened, and that one of the functions also of the institutional biosafety committees is to do risk assessment. So before an approval is done, the applicant must submit a proposal or an application together with risk assessment. So as a consortium, we have recognized this, and last year we did train 10 of our colleagues in Africa, and we are fortunate here that we had Adilson and I think Tien from Burkina Faso. So what has been happening is that qualitative methods have been used for risk assessment, but there is need also for quantitative risk assessment methods, which we have begun, and the discussions are going, and further training is going on in the continent. Then any trainings that we do, we put them in the videos and we put them in the website. So check out our website, you'll be able to see videos on what we discuss. The latest videos were on a GMO training for journalists, which we did the other day, and I can answer questions that somebody had a few minutes ago. Then we have published a points to consider document that takes you through all the requirements from lab through until commercialization of these genetic biocontrol products. Then I mentioned that next week we are going to have an IBC forum in Africa, where Institutional Biosafety Committee Forum, whereby we're going to help them understand the operations and management of institutional biosafety committees. I've been asking people here, but not many people in Africa know about the role of the institutional biosafety committees. So we are going to train participants from countries that have already adopted technology, helping them to develop guidelines, manuals, and even look at decision making and communicating those decisions. I know Tony is going to be naughty. And very soon also we are going to talk about environmentalists. Environmentalists have been people who have been against the technology for many, many years, and because of the laws that exist in many countries. So in April, we are going to bring environmentalists together. They learn about the technology and how to also support decision-making process. Now, the biggest thing that we are going to do this year, which we hope it will be a big success, will be the first global genetic biocontrol conference. This will be the first one. And we hope you're going to register in large numbers. We're going to have a 3-day training, pre-conferences, and 3 full days for conferencing. So if you're interested, I have a brochure here. Check out our website also. And if Tony will permit me and do some marketing, I think I'll be able to give some discount to people from Antifec, which is that in large numbers for this conference. So I do hope that we will work together, and you're most welcome to join us. So one thing that we also have is a mentorship program. We try and bring mentors and mentees together on genetic biocontrol. And Nikolai is one of our mentors. We need more mentors to mentor our African brothers and to work with us to improve on the technology. Then finally, we're working on a registry, whereby we are going to have a portal in our website where we are going to have all the information about what's going on, who's doing what in Africa, what institutions are doing what, who are the individuals like Dear Party. We need more Dear Parties, don't you think so? Why don't you clap for him, please? So, and then, so we welcome you guys to join us. Please take a look at our websites. I brought a brochure also about our genetic biocontrol. So I'm still here for about a few hours, then I'll leave back for Nairobi. So if you have any questions, you're free to ask me. Thank you very much, Tony.
Thank you, Willie. We're a little bit over time. So if there's any burning questions, we can do one while David Mburu comes up. Is David here? There. So Willie, while he's getting up.
Yes.
To Charles's question.
Transponder movements.
No, well, more about the institutional biosafety and efforts that you're doing to coordinate that across different conglomerates of countries like ECOWAS.
Yes.
Can you say anything about that?
Yeah, so NEPAD has been spearheading that. And as a consortium, we are looking at African-wide and starting from institutions. So, so NEPAD has started from Western region, but for us, we are more interested in institutional capacity building. So we have started with 6 countries right now to try and work with them to improve the decision-making process. So Burkina Faso, Uganda, Mali, Ghana, and Tanzania, I think Uganda, yes. Those are the countries we are working with right now, and working in collaboration with NEPAD also to support their activities. Yeah. So Charles Wanji, you are one of the people we are targeting to build capacity in Africa.
Thank you,
very
Willie.
much.
Good afternoon, everyone. Welcome to my topic on leveraging on semi-field system integrated with insectary, animal house, and molecular biology facility, potentially for vector endosymbiont research. And I was probably focusing at the Kenyan coasts. As you know, everyone has a story, and probably nested within our journeys in life. My journey here in Kilifi started in 2010 when I joined Pwani University. And at that time, Pwani was a constituent college of Kenyatta University. So, and being an agricultural university, there wasn't much to do with the like human health research or vector biology. And it happened that while I was still looking for a house, I had happened to have met a training officer at Kemri Welcome Trust, and we were like, can we collaborate? And so the story started. And I hope to take you through the story. So advancement of omics data bioinformatics tool, of course, has made hypothesis testing possible with molecular data, as we probably all know, given that behavioral phenotypic expression are determined at genome level. And of course, there is need to link, laboratory-based and field study, which are very important in identifying mechanism underlying biological systems as well as generating new hypotheses. However, of course, we all probably aware that gaps do exist between data that emanate from laboratory-based studies as well as field studies, especially, for instance, for vector-borne diseases, including malaria. However, semi-field can be artificially brought on board, and like we have been taken through by Diabate, which can help us gap the variation that we observe in the two arms of the data generation. Of course, this has been cited as the best system of mitigating the existing research gap. So at the time, like I've said, the constituent college and Kemri Wellcome Trust, probably they were at very different levels in terms of research focus. So we thought in the conversation that followed, how can we, collaborate and orient probably the key theme of, collaboration, given along the Kenyan coast is full of myriads of, mosquito-borne diseases. So we thought probably establishing semi-field system integrated with insectary, animal house, molecular biology lab, and probably, facility for that could now push the data from sequencing and of course analysis of the data probably would enhance the results of the collaborations. So for instance, also this could also come in terms of infection and immunity studies of vector-borne parasitic interactions along the Kenyan coast, which again was from the university side of life, the focus was different. It was in plans, but how could we bring in the stakeholders? We're working within now the constituent college into vector and health research. So we had our hypothesis that these can definitely work hand in synergy, probably to enhance quality of the results that we generate. So how did we do that? Then we set a couple of meetings where we strategized on how we can push, orient the stakeholders, especially from the constituent college. And we did plan, for instance, to come up with, sorry. We set to come up with infrastructure grant, engage both management at the constituent college at the time and also members, key members of Kemri. So the first grant we got was the Wellcome Trust, which generally fed into setting up the molecular biology lab, state of the art, probably that you may not find in an East African, in a university in East African region. So the next grant came Again, we managed to get a grant from National Research Fund in Kenya that again fed into buying surplus to the molecular biology lab, as well as now setting animal house, greenhouse, for those people who would want to continue with the plant sciences and also crop science, then insectary, and also people who are interested again in marine sciences, course being close to the ocean, kind of trying to bring all the stakeholders and interested parties together. And also now, after we generate the data, how do we move it forward in terms of bioinformatics? The facility, or what we're calling Pwani University Bioscience Research Center infrastructure, again, we discussed with the stakeholders and we saw it wise to disintegrate from being owned by a particular clique of people or interested party. We took it under the research and, extension division, so that in case I have a collaborator and they want to, come and work together, they will just need to come with their finances, and then we are able to, put it through the steering committee. And then, of course, we have a lab manager who look at the activities, day-to-day activities of the lab, and we are able to push forward. So, actually, the management, Pwani University management, gave us the 3 classes actually, which we collapsed, and then we formed the lab. And currently the lab is functional. It's supporting students, PhD student, master student, as well as very brilliant undergraduate students for their projects. If I was also a bit to express a bit on the insectary animal house, the part of the insectary, and then we have the animal house on the other end, and of course the larvae and pupal stage. And now the semi-field is here, so what's remaining is within the 4 compartments of the semi-field, we can now build the huts, experimental huts, and then we can be able to, do the bioassay experiments. So allow me, Tony, to mention that Sam Kinyanjui was very key in the initial talk. Of course, Charles Mbogo was the then acting director, KEMRI, here in Kilifi. And also to mention that the members there also are very key, and even currently very key in terms of steering the function of the lab. So Dan Masiga, most of us probably know him. He came in very handy because we collaborated where we had the Makerere University, Muhimbiri University coming in from the university side, and Pwani University, as well as now the research institutions, including KEMRI Wellcome Trust, for a grant that came in to support, bioinformatics students. In the meantime, like we are waiting for the infection studies probably to stabilize data from the lab, then we can push forward with the bioinformatics side of life. And you can see we also get visitors here. We also get visitors who come in with their projects for a short stint, probably experience within the East African coasts. This is another group that was working with Professor Santi. And the students were from Sweden. Of course, when they do very well, we make sure we cut a cake so that at least they can always remember. So you are engaging your community for future. And of course now, here we had a group photo. So with that, I acknowledge everybody who have, every institution that have supported us. ENBIT, Antivec, ECPE, Fabric, Pwani University, National Research Council, Kemru Welcome Trust, and all of you. And thank you for your ears listening to me.
Thank you very much, David. We're a little bit over time. If there's any burning questions, we can have one, but can the next speaker come up, which is Prince Thabo Mashatola? It's very nice. The insect itself, is it climate controlled or is it open
Yes, it's,
to the
climate
environment?
controlled.
No, it's climate
Yes, you saw the AC.
No, I didn't see it.
It's sorry. Here, this one here.
Okay.
Yeah.
I was wondering if it was
and
that
it,
one. Okay.
even this one, the
Okay.
AC is at the back. Is at the back. Yeah.
Can I start, Tony, or are you going to? Good afternoon, everyone. My name is Thabo Mashatula, and then I'll be presenting to you about the progress we've made so far in my country, South Africa, regarding the sterile insect technique, specifically the development of genetic sexing strains around this technique. And this work, I mean, it's been presented by the fact that malaria remains a global burden, with South Africa not being exempt from this. And I think the previous speaker spoke extensively about this. And the similar issues remain or affect our inability to really eliminate this disease. And this includes insecticide resistance that's developed by so that's developed by the common control methods that we use. So this has called up for use of alternative control methods, and one of the ones that South Africa is exploring is the sterile insect technique. So the idea or concept of the sterile insect technique is not entirely new, as this has been used successfully in mostly in agriculture or against agricultural pests, but in mosquitoes it hasn't like really been used extensively. But what then this concept or this technique involves is mass production of these mosquitoes in a laboratory, followed by sex sorting, then sterilization by radiation of the sex sorted males, and they're released into a target site with the expectation that they will mate with wild mosquitoes, and then when those females lay eggs, the eggs will not hatch. So specifically in this talk, I'll focus on the sex sorting because this is a very critical component of this technique, as what you're aiming for is to not release females, as this can potentially transmit the disease if released. So you want to only release males, as they do not, they do not bite and have no potential to transmit the parasite. So against this, then the work has been ongoing for a while now, and we've explored various sex separation techniques thus far, and this has included mechanical methods where we're trying to exploit the differences in size in the pupae. But unfortunately, and this way has been used in other species like Aedes, for example. But this has proved unsuccessful for Anopheles, so this set us back. But we continued and explored other methods, including spiking blood meals, or through artificial feeding with toxicants such as ivermectin, with the hope that we can target the female feeding mosquitoes. But also with this, there are challenges because, for one, you cannot guarantee that all the females would go on to feed. So this then did not give us 100% elimination. So, and then from this, then the most promising alternative was exploring of the genetic biology of these mosquitoes, and with the hope of developing genetic sexing strains, which would hopefully then give us 100% sex separation. And also with this work, since it's been a while we've been working on this, we've managed to develop a genetic sexing strains for Anopheles sarapiensis that's based on the Dieldrin resistant gene. So originally they managed to induce or translocate the Dieldrin resistant gene onto a Y chromosome of a Sudanese strain, which we then used through a series of outcrosses, managed to also transfer this to our local strain, which retained the South African genomic background. However, while we're excited with this, this strain then, gave us a few limitations, mostly being the fact that the productivity or the hatch rate of the strain was very low. So for SIT, where you need to produce large numbers, this was proving very difficult. And also among this was the possible contamination of the primary strain while working with this Dieldrin insecticide. So then it was proposed that we have to look further into other alternatives. And one of those alternatives was to exploit the temperature sensitive lethality mutation, which has been used or is currently the primary sexing strategy in an agricultural pest called Mediterranean fruit fly. So how this concept works is that the temperature sensitive lethality mutation that they occur randomly in nature, and they were able for this one they were able to successfully isolate this individuals such that when they expose eggs of this strain to a temperature of 34 degrees for 24 hours, they're able to eliminate females but retain males. And then, also just to add, they with this temperature sensitive lethality in medfly, it was associated with the white pupal body mutation. So this for them was very successful because it allows them to, after exposing to temperature, they can go on to use this as a filtering system to make sure that they do not have any recombinants within their strain. So we also try to copy this concept for mosquitoes, and work towards this has been ongoing and is at advanced stages. This includes successfully isolating individuals with temperature sensitivity. So when the individuals are, or the strain that was successfully isolated, when it's exposed to temperature of 41 degrees for 3 hours, you're able to kill off the individuals. But then when they react under normal temperature, they are okay. So so once this was done, then we've been aiming to induce a Y autism translocation of this wild type allele to in such a way that we would be able to use the same technique to kill off females and retain males that we can now release. However, so far there's been no success in in isolating and developing a TSL-based GSS. So this has also called for us to look for other alternatives. And one of them, through work that's been done and pioneered partly by Tony and others for some years back, is the development of transgenic strains for Anopheles mosquitoes. So, and some of this, the work that has been done has been a successful transfer of this or development of this strain into Anopheles arabiensis, whereby a green fluorescent protein is passed on onto the Y chromosome, and sorry, and then through this, and then using, so on the right here, what you see is a Corpus sorting machine. So through this, you can be able to sort out larvae that illuminate the green fluorescent protein, and through a series of outcrossing, you can actually cross the wild type males with females that have a green fluorescent protein, and then end up with a population that's only males but do not carry the fluorescent strain. So that's made possible through sorting with the fluorescent with the Corpus machine. And from this, then we've been able to successfully develop one strain for our South African project, which retained the South African genomic background. And this has excited us a lot because it promises to be a game changer for our project. As you know, the life history trait comparison has shown that this strain is quite is a quite high fitness, at least in a small scale, and shows no recombination in respect to the fluorescent marker. However, we still have challenges in the sense that there's still regulatory approval that needs to be agreed on by our governments through the use of this transgenic strain. So that said, I would like to thank some of these people and some of the funders and institutions that are associated with this work. And thank all of you also for listening.
Thank you, Tabo. So, there's any question? Quickly.
Thank you very much for your presentation. I just wanted to find out if you are planning to extend the work to other species like Funestus, because I saw just Arabiensis and Gambia.
I think from what I know, we already started exploring the use of, or rather investigations into the sterile insect technique in Anopheles funestus, for example. So that, I guess that work is still in the early stages, but the idea is to try the technique in other species and see if it works.
All right, so good afternoon, everybody. I'd like to thank the organizers for inviting me. And today is actually the talk that focuses on target Maria's activities. Then I will have more broad perspectives that cover the views of other GMD developers. And incidentally, it's the last talk I give on behalf of Target Maria. So I'm moving to new grounds, and my new address will be the Swiss TPH, Swiss Tropical and Public Health Institute, and I will be based also at Ifakara Institutes in Tanzania, and I will be hopefully contributing important things to the Transmission Zero project that is based in Tanzania. All right, so going back to Target Maria, you have heard before that this consortium focuses essentially on 3 species of about 800 species present in Africa, and those are the main members of the Anopheles gambier complex that are, if we remove them through suppression methods, potentially we could avert something like 600,000 deaths per year. And the technology that's used by Target Maria and some others is using gene drive. It's very promising and useful technology that allows you to force or to promote some interesting genes and push them through populations so that they inherit it in all progenies instead of just by Mendelian ratios. So the genes that are Target Maria's favorites are genes of fertility, genes that render females sterile or that bias the progeny so that they produce only males. And these approaches can then lead to suppression of target populations. And as Diabate explained earlier, from the onset, Target Maria's vision was to go step by step. And that's not only because gene drive is a novel concept, the end product, what we hope to develop for the future, is a very promising tool. But the very start, just the idea of having a malaria mosquito that is genetically modified was a new concept. So that phased approach allowed to move along some major genetic engineering developments. And then along the way, you design different field studies with the regulators so that you can look at these different technologies step by step. It allows you to do capacity building in many different areas. The regulators build their capacities to assess increasingly complicated constructs. The field entomology part has to adjust their methodology and build capacity to do these field studies properly. Okay, I was probably being over emphatic, that's the reason. So, and finally, stakeholder engagement also has to build up their capacities. So it's probably the most responsible way of doing things, to have this progression in complexity. And it's also what regulators like. Okay, so the microphone is still on, which is interesting. Operates on battery. This is on. It's on here, but not there. It's the projector. Maybe we. break now.
We could just do it on Tuesday afternoon. You're not leaving out.
No, I'm not leaving. But there is a break schedule. Break and then finish it.
Yeah, yeah.
Okay. That's okay, that's fine. Ah, okay.
Okay. Is it coming? Coffee break now. Okay. Yeah. Okay. Change of schedule. We'll have the coffee break now, and we'll reconvene here in 25 minutes. So Fred will start his talk again at 35 minutes past 3. Okay? That's it. Okay.
Great, thank you.
Don't touch anything. Okay. Yes, Fred, please go ahead. After.
All right. So thanks. Thank you for your patience. I will try to be quick and clear, as I hate to keep people away from the poster and drinks. So I was discussing the phased approach in target malaria. And as you might be aware, the first phase has been completed in 2019. So I'll talk a bit about the results of this first field study. With the Burkina Faso field entomology team and other members of the target malaria group, we worked on designing the next step. So if you can read, this is a sterile male self-limiting step, completed as we will see. Second step is a male bias self-limiting strain, and the preparations for that are ongoing, but I'll talk about the design of the field study. And finally, towards the end of the discussion, the talk, I want to talk about self-sustaining, the gene-derived products, and how difficult it is to plan field studies for those, and why we're having broad discussions with all developers and other stakeholders to discuss the potential designs. All right, so the release was conducted in Burkina Faso by Diabate's team at IRSS. These are some images of the release, and it was a small release in terms of numbers of mosquitoes. The journalists thought it was a lot of mosquitoes. For us, it was very little, as you will see. And here are the cages prepared for the release. Some important stakeholders were engaged in the release itself. So much so that at some point we had to say, okay, the release needs to start now. And we went on with it. And the design was quite simple, and it kind of mimicked some of the mark release recapture studies we did profusely beforehand. So the objectives of the studies were to estimate the daily survival of those sterile male mosquitoes, because we knew that from fitness cage studies that they were not so fit. So we wanted to find at least similar results in the field. We wanted to get some data on dispersal within the release village, and from the release village you will see the scale at which we worked later. And of course, for the regulators, they wanted to be absolutely sure that the GM construct had indeed disappeared with the last sterile male dying. So this was it. There was one release on day 0 of the study. It turned out to be early July. And then we recaptured, as for a mark release recapture study, if you're familiar with that, you recapture daily. And then later on, we moved to a monitoring phase where we captured enough mosquitoes monthly to be able to do PCR and to document the disappearance of the GM construct. So in terms of number, the insectary team worked very hard to generate about 6,428 GM males. Because of the way we maintain and cross that strain, we also had access to sibling males, which have exactly the same genetic background, but without the construct. And we thought it'd be relevant to compare those two genetic backgrounds when we release them. So we have those two categories of males. We dusted them all and we released them. And these are the number of mosquitoes that we recaptured from these 15,000, nearly 50,000 dusted males. We use swarm capture, which is a technique very well mastered and developed by IRSS. And we recaptured 463 males. So we're very happy to recapture a lot. We were not quite sure we would recapture any at some point. And then from spray catch in the house, we recaptured quite a few also. But the total was very good and they survived very well and over a very long period of time. We were not expecting to work in the field for over 3 weeks recapturing those dusty mosquitoes. So the recapture rate was quite similar to some of our best rates. Usually the team in Burkina achieves sometimes up to 5% recapture rates. We had 3.5% in this case, given the recapture effort that we used. And importantly, over the period of the study, 15.3% of males we recaptured were the dusted ones we released. So quite a high percentage. Then, of course, all dusted males were PCR analyzed to identify the transgenic ones from the siblings. And these are the numbers. So there was 145 of the GM males and then 382 of their siblings without the genetic construct. And for some of you will already see that, there looks to be lower fitness of the GM mosquito. And this is exactly what we had observed in our cage studies. And if you look at the patterns of recaptures over time, you can see that we recaptured mosquitoes for quite long. The last non-transgenic siblings released were recaptured on day 17 or 18. And then for the GM mosquitoes, we recaptured them a bit. The last ones were recaptured a bit earlier. We reached 0 a bit faster around that day. And the survival rate is lower for the GM mosquitoes. We did quite a variety of things, and if you want to see the details, you can see a poster by Frank that is in the poster session. And the data, actually, we can see here. There should be Nature Communications on there, but it's not appearing. So it's published if you want to see the data. If you want to see all the analysis, you can look at that paper too. But one important phenotype is the male behavior in terms of joining swarms or not, because it gives us some information about in future whether those males will potentially mate with females. We were not really covering mating per se in this design, but we wanted to see if they behave normally. And if you look at the unmarked males, the wild males in swarms, and these are the genetically modified ones and their sibling brothers, you can see that when the team was collecting swarms at that time of the experiment, in the early days, there was a majority of dusted males. So we achieved quite a high release ratio in terms of proportion of males released compared to wild ones. So that's quite interesting, because you could imagine that if we had released many more, actually this sterile strain could have potentially have a small effect on the population size. And then if you look at the swarms and their composition, this is the proportion of swarms which is only wild males and in red, the ones that had mixed genotypes. And this shows us that if it's in red, basically the release males are behaving and joining swarms of wild males in a very natural way. And you can see that following the release, we had a lot of mixed swarms. So females basically had access to GM males and non-GM males, and we hope that they would have liked GM males. So that's good news regarding the behavior. Some important data also that was generated. In terms of the spatial scale, we were recapturing mosquitoes very near the release site inside one village, and also recapturing in the village next door, which is not too far, it's just like about a kilometer. And you can see by the size of these bubbles here, the red bubbles are the GM mosquitoes, orange bubbles, the number of siblings. And you can see that we recaptured quite a few very near the release point. Over time, they spread quite a bit in this village here, mostly within the urban areas where the swarms are found. And then four of the siblings went as far as the next village. No GMs made it that far. So in terms of the distance, Euclidean distance, which is the distance between the recapture point and the release point, there were clear differences because the maximum distance is much higher for the siblings than from the GM mosquitoes, and the maximum for the GMs was 500 meters. The average was 136 meters. And then when we look at previous MRR, this distance is what we found quite regularly for MRR data conducted within villages. So you can expect that within that time scale, which is 3 weeks, we had a distance about 200 meters on average, which is not a big dispersal, but probably it's bigger than for 80s studies, for example. And then, of course, the data was also analyzed by the Oxford team and colleagues, where they fitted a much more explicit model of diffusion and survival. That model also allows to estimate the population size of the target population that's at the time of the release. And it's a maximum likelihood approach. And you can see that the GM survival was actually much lower, like we saw from the recapture rate, much lower than for the siblings. The population size at the time of the release, this is actually per hectare, so it's easier to read the extrapolation here. Population size was probably 28 to 37,000 individuals. Lifespan inferred from there, 2.6 to 3.9 days for the GM males, and then 5.2 to 7.5 days average for the sibling males. And the dispersal is actually a bit larger. It's not significant for the GM males, but because they survive less well, they didn't go as far, they couldn't fly as far as the sibling males. So again, it's a very complicated model, but you can see details of that in the publication. In terms of monitoring, for the regulators, we had proposed to collect 300 mosquitoes, to PCR 300 mosquitoes per month in the following months, until we didn't find any GMs for 3 consecutive months or something like that. And when in the rainy season, we could collect 300 easily, and power calculations told us that this gave us a power of 100% to detect 5% of DSM frequency if it was still in the environment. So we didn't find anything in August. In September, we PCR 300 more and we didn't find anything. And then over time, there was less rain, so we collected less per month. But the cumulative number of mosquitoes that we had PCR'd gave us increasing power to detect any GM if there was any, and we never found any GMs. So with this total number of PCR'd mosquitoes, we had actually the power to detect something like 0.5% of frequency of GM in the population, which is quite sensitive with very high power. The second step, which I will not explain in details because it's kind of confidential, but I'm just going to talk about in very broad line. So the the strain used is called ACPMB1. It's a male bias strain, but this time it's inherited. It's not fully sterile. And because it's inherited, it's quite relevant for regulators and other stakeholders because it will linger in the environment because it's inherited in Mendelian way. It has some small fitness costs and it will disappear a bit slower. And for this, it's going to give us a scale and some monitoring activities that are a bit alike what we would do for a gene drive trial. So it's a very good platform for actually progressing and building capacities in those different areas that are needed when you do a broad scale release. So for those of you familiar with some of the guidelines for trials, the format is alike a control time series with a control village that we track during the course of the study. Then you have a village where the intervention starts, and you can compare the before and after within the intervention village. And it's also quite powerful because you can also control the intervention village versus the control village. So this is not as powerful as a proper cluster trial, but it's a design that's quite strong. So the format is like that. The plan is to have this somewhere between 2024 and 26. And the objectives here this time is to document the persistence of the construct, its disappearance, of course, as before, and to study dispersal. And we're interested, as Penny was explaining, there's a lack of understanding of what happens in the dry season with mosquitoes and what would happen with GM mosquitoes in dry season, where population collapse or go into hiding. So we designed the study to have some idea of what happens in the dry season. We also went for a larger scale. We have about 15 kilometers from left to right here. And in that perimeter of the study, we'll have an intervention village, like I mentioned, a control village. There are other settlements in other directions. So you can imagine that if you sample in all directions, you can do nice spatial studies, understanding the patterns of movement. There is a road passing through, so you can also study whether the road has an influence on dispersal. So this study is not huge. It's a huge amount of work compared to the previous MRR, but it can tell us very nice information about different sources of dispersal. If there is a dominant wind during a part of the study, maybe it will affect the dispersal. The road might have an effect. And anyway, so that's one of the big objectives, to have very nice description of dispersal. And the study will last several months, possibly a bit more than a year, and it will generate this important data on seasonality. And importantly for the team, it's such a big effort and so many samples to process that we had to develop tools that may be used, or they're a bit like the tools we consider perhaps for a gene drive trial, where you have to monitor very large areas. You have to train other teams in neighboring areas to monitor the GM. So we are looking at very practical solutions to detect the transgene and to collect a lot of mosquitoes. So finally, the final step, which is this one that's so interesting, there were good questions by Charles and others talking about cross-boundary movements, and I've had long discussions with the team in Burkina. And within Target Area, we have long discussion, and there are discussion groups with FNIH discussing that. So here are some aspects and important issues and points of discussion. The so-called product one, which is this dreamlike gene drive. You don't need a lot of mosquitoes to transform the whole of the population of your target mosquitoes, and it will resolve at low cost the malaria issue. Basically, that potential implies that in your target product profile, which is a bit your selling pitch, you have very attractive characteristics. And usually what we hope will be a product one, the real vector control tool, is a low threshold gene drive. In other words, it requires few mosquitoes, and these few mosquitoes can spread their genes into the population until the gene reaches fixation. And in our case, the population would then crash. So that's a wanted characteristics, but as you will see, it comes with a small cost. If you can do that, you can target endophilic and exophilic Anopheles coludzii gambiae populations, which is immensely useful. You can target populations that are difficult to access. In the rural areas, that's very useful too. And if it works well, also because it's spread so well, it will cost hopefully less than bednets and IRS because you need to sell that also. So in terms of drives, there's this relationship between the frequency at which you release the GM mosquitoes, their fitness, and then the likelihood that the drive will, the gene of interest will reach fixation. So we are trying for product one, usually the aim is to go for something that can reach fixation whilst you release few of them, because that's what makes it so effective and such a nice concept. But on the other hand, it comes with the costs. And here you have some simulations very like what Penny showed. Imagine Burkina Faso here, and at time, we are negative, we are before the release. This is a simulation by the Oxford team, and now we release in 5 villages, 100 kilometers apart, and you will see the spread of the construct. You have seasonality, population disappear and reappear, some remain along the rivers, and you see over time that the red populations, the populations where there is fixation of the transgene and therefore they are crashing and disappearing, they spread spatially, yeah? So I'm gonna stop, sorry. I don't know how to stop this. Anyway, but the message here is that because this GM mosquito spread over time, if you imagine a structure for a trial that is like a bednet trial, for a bednet trial, we could start with our 5 clusters without a certain bednet, and then 5 clusters with bednet, and you would have stability of this distribution over time. But with the GM mosquitoes, those clusters will start intermixing. It's called cluster contamination, and that affects the scale of the study you design, because you want to keep your control clusters or non-intervention clusters clean, and you want your intervention clusters clean, and you don't want to mix the two. But with this gene drive, and the low threshold one in particular, there is rapid spread, so you need your clusters to be well spread in space. So the question is how spread? Because we also know from other studies, and this is work by Tovi Lehmann, that we suspect there's also long distance dispersal that nobody quite knew about, which can be mosquitoes being caught in dominant winds in high altitude and moving between borders sometimes. And if you read this paper, which is very stimulating, that was published in Nature in 2019, by capturing mosquitoes caught in nets in high altitude on sticky nets, the group by Tovi kind of inferred where they came from with the dominant wind on that day. And you can see that some of the trajectories are very long. And therefore, if there's mosquitoes moving so far, you can have transgenics moving far. And then if you have a cluster where you had fixation of the GM and the population disappeared, you can have recolonization of this vacant niche by incoming mosquitoes. So it has a huge effect on simulation models and on the size of these possible interventions. So some of the discussions pertain to what do we need to do to understand migration. Now, in terms of framework, specialists sit down to try to write guidelines about how to design those trials. And this is a paper that was published in 2018 after a consultation of experts like some of us. And in that pathway, I don't know if you can read, but they advocate that for trials towards gene drives, there should be large-scale studies, indoor, outdoor maybe, small-scale isolated releases. So how can you do that given that there's a lot of dispersal? That's one question. And then the recommendation was small-scale open releases followed by large-scale open releases, and then post-implementation. And you realize that because this product one can spread so well from a few dispersing individuals, these 2 steps are not very discrete. So that's an issue. And then these guidelines that are a bit more recent kind of covered up for that by calling these 2 steps and merging them stage open field releases. Okay, so it sounds good, but how would you implement a stage open release? Yeah. Very difficult to move from a small open release when you think about all the logistics, the regulatory permission, and then this moves by itself to a large scale open releases. So that's one of the major challenges. So I'm going to accelerate. So the problem is that you might want to start with a cluster trial, which is a very powerful statistically approach, but you need to have your clusters very separated, and still they might merge very quickly, and you'll end up at best with a kind of control intervention comparison where you can't really predict where the next intervention area will be because of this long dispersal. So that's one of the big challenges we have when designing that. So if you have that scenario, you're going to propose to governments, to regulators, and to the broad public an open-ended task, and they need to trust you that you can do that. The simulation I showed you already involves, besides Burkina Faso, Mali, Niger, Ivory Coast, Ghana, Togo, Benin, as this trial will spread. So in terms of regulatory framework, it's very difficult to have everything in place to be able to ask the permission to do so, and then to actually get the permission when you cannot really explain where this thing is going to stop. Then you need to find funders that will accept to fund this open-ended trial. And then you need to have a model engagement that can cope with this. So the challenges are really huge. In terms of African regional governance, it needs to be in place, and there are efforts being made to get that in place. And then one of the elephants in the room is that when you've convinced everybody, perhaps, that this product is great and safe, and that we should indeed do this trial that might basically spread through different countries, then you say, but actually, we think that resistance will arise within 2 years, but we don't know where and when. So that's another problem, and you can see how the task is difficult. So what can we do, of course, and that's a debate I hope we can discuss together at some point, and everybody should contribute somehow to these discussions. I think it's important, and this is not Target Malaria speaking, it's my own opinion. I think it's important to really focus on these confined studies. If we can do any, we need to make them well. If we can do large isolated studies, we should do them, because they need to set the stage for these hugely complex and difficult decisions. They can enable things. I think we should understand and use what we know about the evolutionary resistance for low threshold drives to try to be able to predict those, or even engineer a shelf life of the gene drive so that we can explain, this thing is going to be active for that many months, and then therefore after that we expect that it will die out. So that would be one way of going about things. We need to understand the dry season and dispersal dynamics better. And then we could possibly look into higher threshold drives, and I was very happy to hear 2 talks this morning that are not the low threshold drives, that are intermediate so-called products that will not spread that fast. Because I think that it may actually bring some solutions as we think about this step, which is very complicated. And we might actually need some intermediate steps like that. And then finally, there is genetic confinement. And if you could, for example, like Tony was hinting at, only target some genotypes of the mosquitoes, only the resistant ones, and only transform that or kill those, that would be another form of isolation that would be very appealing. So with that, I will tend to finish. There are other aspects I'm interested in terms of advocacy, like Diabate and many others. I think that African researchers should lead a lot of that innovation. It will help convincing governments and gathering support for that. So innovation in Africa, genetic engineering should be there, and the governance should be in place there also. Thank you very much for your attention.
Thank you, Fred. You got some extra minutes because of the disruption. Question there at the back from Charles. Anyone else?
Thanks, Fred, for the nice presentation.
Go ahead.
Yes, just to maybe ask you to elaborate on potential impact of population, repopulation after the wet season. You remember that work from Fred Sima in Senegal, showing that after the rain season, immediately the local population that seems to be extinct was immediately repopulated with the same genetic background as a population 300 kilometers apart. So what will be the impact of that in relation to the point of release of your GMO mosquitoes? Secondly, what could be the impact of chromosomal inversion, also chromosomal forms, which can be barrier to gene flow among your population? So in terms of the modified mosquitoes, are you taking that into account? Thank you.
All right. So in terms of the high likelihood of dispersal, I think that fits into the envelope of what Tovi Lehmann's study shows. We have to take into account the likelihood that some of those populations are recolonized by moving mosquitoes, almost migratory mosquitoes. And the modelers can do that. The difficulty is to know how much of it, when, and at what time of the year they can accommodate some of those questions. And I think there are efforts to do that, or to at least have in your different dimensions and parameter space to have room for those scenarios. So that work is being done. In terms of the chromosome inversions, there can be opportunities to target some subpopulation of mosquitoes as a genetic barrier. That's not been explored that much. But in terms of developing GM mosquitoes and backcrossing them into the local background, which is something regulators insist on, as a project, you will need to look at the frequency of some major inversions, and you'll want to know where your construct is with relation to those chromosome inversions. Now, if it's away from an inversion, then you don't necessarily need to look at every possible inversion, because as you know, there are many, but you need to make sure they are not located in one of the inversions, that's for sure. So that I think anybody working on Anopheles gambier and backcrossing strains to the local strain will have to keep an eye on the major inversions.
Okay, we have one question, but just on the inversions actually, because I work on that. The inversions don't make much of a difference to the gene drive copying. So as a protective resistant mechanism, it's not necessarily envisaged that that would be adaptive. We'll have one question from Anna, just to mention that and then Thabo, if there's time, the bus is scheduled for 4:30 for those going to the other hotel. Correct, Michelle? Yeah. Did you hear that second bit? It will come back from there at 6 p.m. to come here for the poster session. Anna?
Thanks, Frederic. A quick question. For the PMB1 field study, are you planning to include population suppression as an objective? Because I would imagine for that you'd have to do a large release or multiple releases. Thanks.
So there's different people who were involved in that discussion, Frank, Diabate and others. And actually Ace from Oxford also generated interesting results that gave us a more informed. So we would have the ability of detecting a suppressive effect of those punctual releases if we wanted to. If they occur, we'll be able to tell. We'll have enough power for that, but it's not as a main objective, because otherwise you set the expectations that are very high. And truly, we looked at different scenarios where this would be our main objective, and it was going to be likely difficult to achieve. So we are having a more like a conservative standpoint.
Thabo?
Thank you for that very interesting talk. I just wanted to ask regarding the MRR. I just wanted to maybe find out from you how you were able to tell the difference between the mosquitoes you released and the native ones, or rather, what did you use to mark the mosquitoes? And do you, by any chance, know if this had an effect on their survival subsequently?
Okay, so we use the old-fashioned fluorescent powder, dusting, and like I said, we have a lot of I mean, the team in Burkina compared colors of powders, did a lot of fitness studies. So we know that the best color is red, and then yellow is very good too. Blue is for some reason not very good. So that was the method of choice, mostly because we had huge experience with that. And yes, so that's the easy answer.
Okay. Thanks to all the speakers in this afternoon session. Sorry for the disruption. I told you the bus times. The poster session is at 6:30 around the lunch area, and that's scheduled for an hour. So please do attend, because people who brought those posters from very far away put a lot of effort into them. And dinner is scheduled for 7:30, immediately after the poster session.